Thanks, David. I'd like to begin by simply thanking the Center for the invitation to speak about our research today. But I should also begin with a bit of a disclosure. I am the founder, or was a founder, SAB member, shareholder of Proteasome, which was purchased 2 years ago by Onyx Pharmaceuticals, and I continue to be a consultant for Onyx. I will be showing data generated by both Proteasome and Onyx today, and I should also mention that despite that I'm showing the data, I do not in any way represent Onyx in my presentation. And so my lab has been working for the last 17 years at the interface of chemistry and biology using chemical approaches to address different aspects of cell biology. And this has allowed us to generate a lot of different probes, but it also has allowed us, we feel, to address a challenge today that's being faced by the pharmaceutical industry, and that is, given the fact that we know every single possible drug target out there, how do we focus on those particular compound or those particular proteins that, when inhibited with a small molecule, would be pharmaceutically vulnerable? And so this challenge of target identification and validation is an important one. And the approach that we've been taking is actually to look to Mother Nature. The chemical diversity that is offered in nature is quite immense. And what we try to do is to combine chemistry and biology to explore the mechanism of action of biologically active compounds with the goal of trying to not only find out their target protein and hopefully new tools for cell biology, but also to identify possible new targets for drug development. And this is a strategy that has been successful in our lab for a variety of different natural products and identification of a variety of different targets. Methionine aminopeptidase is the target for the natural product Fumagillin and several others. But what I'd like to do today is talk about one particular natural product that was isolated from this microbial source by Bristol Myers Squibb Research Institute in Tokyo. This was a natural product-based institute looking for anti-tumor, anti-solid tumor activity in extracts of microbial broths. And one compound turned out to be very, very potent in their assay. It's a peptide epoxy ketone that goes by the name of epoximycin. It had been shown that this epoxide was absolutely critical for its biological activity. And so, given the fact that this had potent activity and had a potential way of covalently interacting with its target protein, we set out to try to find out how it worked. And that, of course, meant for synthesizing it, since there was no commercial or private source of epoximycin at the time. But the total synthesis of this natural product also afforded us the ability to make an affinity reagent, a bioaccumulated form of epoximycin, with which we went hunting. And so you're looking at a blot of bioaccumulated lysates that have been treated with this affinity reagent in increasing concentrations and simply probed with avidin HRP. And as you can see, there are two major bands here, both of which disappear if one pre-treats the cells with excess amounts of the natural product itself, indicating that this interaction is a specific one between the natural product and these receptors. And so what we found was that these two proteins are catalytic subunits of the proteasome, which, as you know, is the major source of mechanism for degradation of intracellular proteins. It's a very abundant protein complex. It makes up 1% of intracellular proteins. And it's highly conserved. But what is of particular interest to this audience, I would imagine, is the fact that this is the primary target for an FDA-approved drug known as Velcade or Proteasome. And this is the only small molecule that currently is approved for use that targets the proteasome. This is the 26S proteasome, meaning that it has the central core cylinder known as the 20S, as well as two regulatory caps on the end. These regulatory caps are responsible for recognizing ubiquitinated proteins, removing the ubiquitin, recycling it, and threading in the protein to be degraded into the cylinder where there are these inner rings here, the beta rings, have three different catalytic subunits that are defined by their preference for cleavage after a particular amino acid. These are named the caspase-like site, the chymotrypsin-like site, and the trypsin-like site. Now, in addition to the constitutive proteasome, there is an inducible proteasome that is restricted in immune cells and is induced by gamma interferon. The point here is that these catalytic sites are swapped out upon gamma interferon induction, such that these now have slightly different cleavage specificities that will allow for a better peptide repertoire to be generated and presented on image class 1 molecules on the surface of the cell to facilitate immune cell signaling. I'm going to be talking about inhibitors of the constitutive proteasome, but there is a lot of interest in developing immunoproteasome-specific inhibitors for autoinflammatory diseases such as rheumatoid arthritis. Now, when we found that this class of small molecules, this natural product, binds to, and I'll show in a second, inhibits the proteasome, we looked into the literature to determine what other proteins behave similarly. And it wasn't too surprising, now that we look back on it, that our compound inhibits the proteasome, because these other proteasome inhibitors all have common characteristics in the sense that they are peptide-like for the most part, with a electrophilic warhead, either an aldehyde or a vinyl sulfone or a glyoxyl. Now, the exception to this is a natural product lactocystin, which was considered to be the gold standard in terms of proteasome inhibition, which spontaneously rearranges to give this beta-lactone, and it's this molecule that is the inhibitor of the proteasome. But up here, I also point out again our friend Velcade, bortezomib. It is a dipeptide boronate as a warhead, and that boronate will be important in a second. Now, using different peptides that when cleaved will fluoresce, and these different peptides having different amino acids at the P1 position that would allow us to sample these three different subunits, we were able to determine that our natural product was a very potent inhibitor of this chymotrypsin-like site. And this was important because yeast geneticists had already shown that inhibition of the chymotrypsin-like site relative to these other ones was absolutely critical for life. You can inhibit these without having a serious impact on the cell cycle. This one causes cell death. And as you can see, again, our natural product compared very favorably with the gold standard in terms of the beta-lactone. What our compound did not do, however, was cross-inhibit a variety of other proteases. I mentioned in a previous slide that there were a number of peptide, aldehydes, and glyoxyls, and other so-called proteasome inhibitors. They do inhibit the proteasome, but they were originally, or their classes were originally identified as inhibitors of general proteases. And therefore, at high enough concentrations, these so-called proteasome inhibitors will cross-react with a variety of intracellular proteases. That really was surprising to us that our compound did not inhibit. And it wasn't until we got a crystal structure of our compound bound to the 20S proteasome that we were able to understand why we had such a specific inhibitor of the proteasome. And I should back up and just describe a little bit about the biology of the proteasome. These catalytic subunits should not be, or Mother Nature does not want these catalytic subunits to be active when they're not part of the cylinder. They don't want to have unregulated proteolysis. So they're made as longer proproteins that are inactive that undergo autoproteolytic cleavage upon assembly into the cylinder. And as that cleavage will generate, obviously, a new amino terminus, and as that amino terminus, that first threonine, is the active site. And so, if you'll bear with me here again is that first threonine on the proteasome. If you can imagine this not being an inhibitor, but instead being just a peptide, a substrate, and that this now being an amide bond here, that hydroxyl on the side chain of that first threonine attacks the carbonyl here. It goes through this intermediate. Water would normally come in here, and you get hydrolysis generating two different peptide products. But what we think is happening is that when we now have a peptide with an epoxy ketone pharmacophore, you still get that first attack. You still get this intermediate. But what you see is this is a flat, planar geometry here, sp2 hybridization of this carbonyl. This is not. And what that means is that this geometry now forces that carbon in a different position than it was here. And so we believe that when this is in this position, there is a second nucleophilic attack by the amino terminus itself. And so what you end up having is a closed ring structure, a morpholino ring analog, that results from this bidentate, this double nucleophilic attack across this pharmacophore. Obviously, other proteases have nucleophiles at their active site. What makes the proteasome specific, what makes the proteasome different, is that its active site is the very first amino acid. And that allows for both the side chain as well as the amino terminus of the protein itself to be able to attack across this double nucleophile. And so that's why we feel we have a very specific proteasome-specific pharmacophore. And so what we set out to do next was some medicinal chemistry. Could we improve upon Mother Nature? Could we vary these residues to come up with something that was possibly even more selective for just the chymotrypsin-like activity and not touch the other ones? You remember there were 2 bands on our affinity gel. And could we also increase the potency? And so we set out to simplify things, again keeping the pharmacophore the same. We simplified things and just put leucine, 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 leucine. Tetraleucine, trileucine, dileucine. Now remember, the Velcade is a dipeptide. We did not see any activity. We only were able to see significant activity when we had 4 amino acids. We now wanted to vary each position individually, putting in increasing steric bulk. As you can see, increasing steric bulk gave increasing activity. I'll quickly go through these. We systematically looked at P3, 4, and 2, again, steric bulk. And finally, looking at this position here, we found an optimal. When we took all of these data and condensed them into a consensus amino acid, we came up with a molecule we call YU101. And that this is a molecule that had increased potency as well as selectivity and was many-fold better than Velcade in terms of its ability to inhibit the proteasome. At this point, we, with the help of Tommy Chen's lab, looked at this in vivo activity in terms of xenograft tumor studies and showed that, yes, this still retained biological activity. And so together with Yale's help, we started a company called Proteolix that first order of business was to take this compound that which we had not designed as a drug and therefore had very, very poor solubility. Yes, it could be dissolved in DMSO, but you can't inject patients with DMSO. The first thing that Proteolix did, ironically enough, was to add a morpholino group on this end of the molecule to increase the solubility. And that increased it to the point where now this is the drug candidate Carfilzomib that has been in clinical trials now. And as I said, we were able to take this through phase 2 clinical trials. Onyx bought Proteolix 2 years ago and is completing the phase 3 clinical trials now. And so some of the biology, it's nice to be covalent. So if you look at just simply the chymotrypsin-like activity, you can pulse treat cells with these 2 different proteasome inhibitors. And as you can see, bortezomib, which is not covalent, you get a rebound in proteasome activity. That is not the case with carfilzomib. And this is simply reflects that this increase reflects the resynthesis of the proteasome. And this is reflected also in cell viability. If you look at, again, a 1-hour pulsing with carfilzomib versus bortezomib, that you need higher concentrations of bortezomib when you treat with only a single dose relative to carfilzomib to get the same level of cytotoxic activity. What we found particularly encouraging is that one could take cells that were resistant to Velcade, bortezomib, in this case for multiple myeloma patients, and they still responded to carfilzomib. And in fact, one could take a human cell line that was sensitive, shown here in black, to bortezomib, and by treating it for several weeks in the presence of bortezomib, make or select out a resistant cell line that in red still responded to carfilzomib. So even though they both inhibit the proteasome, there still was the hope that carfilzomib could be useful for patients that had already seen bortezomib. Now the big problem with this, and I think that many of you know this that have used bortezomib, is that there's a peripheral neurotoxicity that's associated with Velcade. A significant fraction, up to 30% of the patients, have a tingling or a pain in their extremities, their hands and their feet, some to the point where they will stop taking the drug simply because the pain is too high. And so this peripheral neurotoxicity was of great concern to us because we were not sure whether it was on target, meaning that all proteasome inhibitors would have this, or whether it would be off target. And so what you're looking at here is just a cell culture of neuron-like cells that, again, you can see bortezomib over extended treatment kills these neurons, that carfilzomib does not have that problem. And so the group at Proteolix was able to determine that this had to do with a particular serine protease, mitochondrial protease, HTRA2, that is upregulated when you inhibit the proteasomes, but despite the fact that it's upregulated in neurons, that is absolutely required for the survival, and that bortezomib inhibits the activity of this. Again, highlighting the fact that bortezomib inhibits the proteasome, but also hits other proteases. And this happens to be a neuron-specific protease required for survival. And so we believe this is one reason, or I should say carfilzomib's inability to inhibit HTRA2 it explains why we don't see the peripheral neurotoxicity. So where are we with this? We have a nice drug candidate, peptide-like in nature, a unique proteasome pharmacophore. Sustained inhibition results in apoptosis in a variety of different tumor cells. There is no neurotoxicity, as well as there is a activity in xenografts that were already resistant to bortezomib. So I just want to briefly give an outline of the a summary, if you will, of the clinical development of this. To date, 11 clinical trials have been started. 9 are completed or are near completion. Over 1,000 patients have been treated as of last summer. Primarily, we're looking at multiple myeloma, non-Hodgkin's lymphoma, but I'll briefly touch on this potential for solid tumors. And now there are two phase 3 open trials. I won't go through this in great detail because this is published, but suffice it to say that we were able to see that a stepping up, a dose starting at a lower dose and then increasing the dose, was a significant advance in how we designed the trials. These two trials, there were two second phase 2 trials. This is the trial in which we're asking for approval for the FDA. And so this trial was looking at relapsed and refractory multiple myeloma cells that had at least 2 prior treatments, one of which had to be bortezomib. And there was a partial response rate of 18% in here. Now, there was tumor lysis syndrome in these, and so there was a modification to this trial, 003A, in which now there was a prophylaxis introduced to address this tumor lysis syndrome. And again, the sample size was increased, and we were able to see something, again, in moderate, if you will, partial response. Now, the second phase 2 clinical trial was open to patients that were bortezomib naive. And this was absolutely critical because we wanted to see what would this behave, how would this compound behave in that patient population. And you see, not surprisingly, that if the patients had not seen bortezomib previously, that our partial response rate was increased. So to summarize these phase 2 clinical trials, this is a robust and durable single agent that if you look at relapsed and refractory naive, bortezomib naive patients, you're getting an overall response rate over 50%. The tolerability profile was attractive, although there were still some common adverse events, anemia, thrombocytopenia, and fatigue. Most importantly, we did not see any peripheral neuropathy, suggesting that this would be favorable for combination therapy. And so where things stand is that there is a phase 3 clinical trial, in this case in combination, Carfilzomib with Revlimid and dexamethasone, and so that this is ongoing. But I just want to finish up by mentioning some potential applications in solid tumors. Because solid tumor cells certainly require longer proteasome inhibition to undergo apoptosis. And so the challenge is going to be how can we increase the exposure of the drug to the patients to get that higher level of proteasome inhibition. And we now know that by, instead of a single bolus, if one administers this over a longer period of time as an infusion, that one can now get up to 95% of the proteasome inside the cell inhibited. And so what that's telling us is that in some of the solid tumor patients that have been tested, we are beginning to see some responses. And this simply shows a renal cell carcinoma patient before treatment and after 10 months on Carfilzomib. So we feel that there is some hope there. So to summarize, we've been able to show that this is a potent and prolonged inhibition, a specific inhibition of the proteasome with Carfilzomib. As a single agent, we see activity in patients that have been pre-treated with bortezomib. The Revlimid data, in which we couple with dexamethasone and Carfilzomib, seems to be very encouraging, and we hope to be able to see more data when the phase 3 data are available. And so finally, we feel that this profile, both the safety and activity profile, offer unique opportunity to really maximize the use of proteasome inhibition, not only in multiple myeloma, but also potentially also in solid tumors. And so with that, I'd like to just acknowledge the talented group of chemists and biologists, my collaborators, and thank you for your attention.